process of preparing your laboratory for accreditation improved in some measure the work in the laboratory?
I don't think it necessarily improved our work product. I think what it did was to bring it all together for us, and I think it addressed some issues. I mean, when you talk about accreditation, this would even include safety issues, evidence handling issues as far as security of the laboratory. All those things are part of it. I don't think in a particular way that it changed our work product. I don't think it changed the quality of our product. I think what it helps to do is to tell the users of our services that we are meeting certain minimum standards, and so it's saying that we meet those standards.
I'm not asking you, I think you were asked on direct examination, were your results as reliable before the accreditation process as after? You recall that?
Yes.
That misstates the testimony.
Oh, not asking you that question.
Okay.
I mean, in terms of cases, are you aware of making a mistake in any of your forensic DNA typing cases to this point?
No, I'm not aware that I've made a mistake in any of my cases.
Right. On the other hand, would you agree that the process of accreditation, which I think you've indicated, you review all the different techniques in your laboratory?
Yes.
You look at how various people in the laboratory handle biological evidence.
Yes.
You would look into some of the factors that we discussed this afternoon in terms of trying to prevent cross-contamination and precautions that ought to be taken.
Yes.
That's the kind of thing that's done during the accreditation process.
Yes.
And during the accreditation process, did you work on that manual that we've talked about?
Yes, I think we had to put together the manual as part of our accreditation, yes.
And in putting together the manual, you went through the details of exactly what people did,
Yes.
and people learned exactly what procedures ought to be followed with care.
Well, people didn't learn that from the manual. We knew those things, and so we incorporated those things into our manual to say that as a reference that this is how we're doing things, and we're telling people that this is how we're doing things.
Right. And maybe some people in the laboratory had one way of doing things and other people had others?
Well, we were actually pretty much all on the same wavelength as far as those concerns, but there were some minor things, yes, that we improved upon.
And in the process of accreditation and in the putting together of this manual, what happens is that you set out uniform standards that you can be sure everyone associated with that crime lab will follow.
Well, I can be sure that they know that they're supposed to follow those, yes.
Right. And you would agree that that's a benefit of going through the accreditation process?
Yes, I would agree with that.
Now, on direct examination, there was some discussion of your role in what's known as certification.
Yes.
And accreditation is something that's done for the whole laboratory, correct?
Yes.
But certification is a process where an individual criminalist is examined.
That's correct.
And I think you told us something about being on the board of American, what is the title of that?
I was on the board of examiners that, for example, I would work on certain test questions. I would also proctor the examination.
Mm-hmm. And this would be for the individual analyst, the certification test.
Yes.
And would you agree that the process of preparing for the test has the effect of improving the work of an individual analyst?
Yes, I think that's a fair statement. In other words, part of that is the educational benefit that one gains from preparing for that sort of examination. Yes.
I mean, no matter how experienced one is, you can always learn some new technique that will improve your level of practice.
Yes,
So you would agree it's important that criminalists who work for a number of years in the field be certified?
I think it's important that the criminalists move towards certification, yes. Our field has been slow to do that, and we are now coming online with certification more and more.
Do you know Dennis Fung?
I know him from watching him in this case, yes.
Do you know if your organization has certified him through its examinations?
I don't.
It's irrelevant, beyond the scope.
Sustained.
Are you familiar with the guidelines for the collection of DNA evidence that was published by the FBI?
Yes, I think, was that the one that Dr. Lee was a co-author of? Yes,
All right. Have you reviewed that document?
yes.
All right. Do you rely on it?
Objection, it's vague, rely on what, for
Sustained.
what reason?
All right. Is there anything you saw in that document you don't rely on?
Objection, still vague for this
For
statement.
purposes of methods that you, in your expert opinion, would believe are sound for the collection of evidence items for purposes of forensic DNA analysis?
I don't recall any statement in there that I disagreed with, no.
Now, the last part of this section of our discussion here, Mr. Sims. I have a series of photographs, Your Honor, that I'd like to mark.
1160 A through,
Yeah, maybe 1168, A, B, C, D, E, and F.
all right, 1160 A
And
through.
you and I reviewed these photographs, did we not?
Yes, we looked at them just briefly.
And these are photographs of you in your laboratory.
Yes, these were taken a few weeks ago by Dr. Blake.
And these photographs show you at different stages of the process by which you, I guess, initially handled and cut the samples in this case.
Yes, Dr. Blake asked me to reconstruct and just sort of go through what it looked like as I was processing the samples in this case.
And could you review those and suggest which order we ought to go through them?
I mean, you didn't put those in order, A through?
I think we did, but I'm only asking him whether I have the order right. No, I got it.
How about if we renumber them in the order we're going to use them? Renumber them, re-letter them in the order we're using
Yeah,
them.
I'm going to do that.
All right.
Let me show you B.
Okay.
We're going to start with B.
Start with B. A is a surprise. Could you describe for us what's depicted in A?
Yes, those are, I believe, my hands. What I'm showing is the process of how I'm cutting out a sample here. This is just a blank card, obviously, it's a white card. I'm showing that I have a laboratory bench that I have bleached now. I have
Well,
a,
let's just stop right there. The laboratory bench is the bench that blue, what would you call it, hospital?
hospital-type diaper, we call them, something
Okay,
like that.
hospital-type diaper, that's what it's on, correct?
That's correct.
All right. And so before you put that hospital-type diaper on the bench, you bleach it.
Yes,
And the purpose of bleaching it is to make sure that you get rid of biological material that could contain DNA.
yes,
Then you put the hospital-type blue, what should we call it, blanket,
sure.
on the bench.
Okay,
Then what's that white paper on top of the blue hospital blanket?
okay. I would, the way I'm processing this case, the way I process this case, and other cases certainly too, I would put down a new one of these blue pads for each one of the items that I looked at. Keep in mind that each item is, generally, each item is a coin envelope that contains 2 bindles. So inside this coin envelope are 2 bindles. One of the bindles would contain the swatch with the blood stain on it. The other bindle would contain the substrate control. So I would change this blue pad between each one of those coin envelopes. So now, when I would open the coin envelope, I'm sorry, before I would open the coin envelope, there would also be a one of these white Kim wipes. It's just a piece of white tissue, a large size tissue, like a Kleenex somewhat.
Is that the white,
Yes,
Chem wipe is the white, large white object on top, larger white object on top of the blue blanket?
yes. And so I would process the bindle containing the stain on top of one Chem wipe, and then I would change the Chem wipe and then process the next bindle containing the substrate control with a new Chem wipe.
So what you're actually doing there is that just for one item, all right, one item that contains the swatch with the specimen on it and the substrate control, between the substrate control and the specimen, you would change the surface you were working on, that Chem wipe.
Yes, in most cases, I think on some of these there may have been, it may have just been one Chem wipe, but I'm working on, you can see it's a large Chem wipe, so I'd work on 2 different areas of it.
But either different areas or you change the Chem wipe.
Yes,
All right, what would be, and the manila envelope there, that's to replicate the coin envelope?
yes, that's to replicate the coin envelope.
All right, and on the upper left-hand, well, I guess it's the upper right-hand corner of the screen, we see another one of those blue hospital blankets.
Yes,
And what's on that?
excuse me. Well, that would now contain that orange object is a test tube rack. And so as I process these test tubes, I would place each tube individually into that rack. In other words, once you put a stain swatch into a test tube, then it's capped and placed into that rack so it's out of the way of the other material. And then also on there, you can see there's another little Chem wipe that has a little blue plastic tube popper on it. That's used to pop these tubes open so that way you don't do it with your glove, you do it with the tube popper. And then finally, I think I'm laying out another Chem wipe that would have the capped unused tubes. In other words, where I would get the new tubes from, because those come out of an autoclave jar at one point, and then you would lay some of those out capped, ready to go.
I'm sorry, finished? Just use the word autoclave. Could you tell the jury what an autoclave is?
Yes, it's a process whereby reagents and equipment is steam sterilized. In other words, it takes things up to a very high temperature to sterilize.
And the purpose of that is?
Well, the purpose of that is to destroy biological material and to break DNA down into very small, very small fragments.
Helps prevent cross-contamination.
Yes.
Okay, now let's go back to you say a scissors in your hand,
Yes,
and that's in your right hand, what's in your left hand?
that would be my forceps.
All right, and you were asked some questions on direct examination about your forceps. Sometimes people call them tweezers, or like
Yes,
tweezers.
tweezers.
Now, are your tweezers serrated?
No.
They're like the tweezers that, or the forceps that jewelers use. They're ground down, correct?
Yes, they're like those.
All right. And it's your understanding that forensic DNA scientists that do the work you do will employ such ground down forceps.
Yes, that's not just DNA, that's from conventional serology days.
Conventional serology days.
Yes.
It's a general practice among criminalists to avoid handling samples with serrated tweezers, because the problem with serrated tweezers is that they're harder to clean.
They are harder to clean, yes.
Okay, so please go on and tell us about what you're doing there.
Well, I'm just proceeding to cut into this sample is what I would be doing. And I think that's it.
Okay, great. Let's move on to C.
Okay?
What are you doing here?
There, you can see the tap is running, and I'm washing my forceps.
Or what are you washing them in at that point?
That's just tap water. In other words, that's a big gush of water to get any of the bulk material off.
All right, so you start with the water to get the bulk material off. Is that correct?
That's correct.
Let's move to D. What are you doing in D?
I'm wiping my forceps with a Kim wipe.
What are you doing in E?
Now, at this point, this is the next part of the cleaning process where I'm actually rinsing them down with a little spray bottle of alcohol.
And what are you doing in F?
This is the flaming process. Whenever that Bunsen burner was lit, people in the laboratory knew that Dr. Blake and I were working together. But that is used to what we call fire or flame the actual tools, and that's sort of a final, ultimate killer of anything. I mean, it would certainly carbonize any biological material that may be left over.
And you would do this between each item.
Yes, and that would be even between each substrate control, of course, too.
You go from the specimen, and you would go through this cleaning procedure?
Yes.
Including the last part of the autoclaving or using the Bunsen burner?
Yes,
And then you would move to the specimen, or specimen substrate control, you clean in between.
yes.
And the last picture, which I know you didn't want me to show, that's you in the lab, right?
Yes, this is a picture of me in the laboratory. That's my U.
Okay, thank you.
That's your U?
Yes, the shape of the bench is a U, so we call those U's.
What's our timing? I can't even remember. Did we take a break, or we're moving right through it?
We're just
I forgot if we took
moving
a
along,
break.
4:30.
Okay. Okay. Let's now move to the Bundy blood drops.
Okay.
Now, plastic bags. In your opinion, sir, putting a wet swatch, or swatches, in a plastic bag, folds over, then putting that plastic bag in a coin envelope, putting it on the floor of a crime scene truck for 7 hours in the month of June. I won't say anything about, just make assumptions about the temperature being something on the order of in the 60s in Brentwood.
I
In
grew
Los
up in
Angeles.
LA, so I know
Okay.
what June is like. It's usually a little overcast part of the morning, and it's
Well,
better
why don't
sun.
we assume it's about 11 to 12 o'clock in the afternoon?
Okay, the skies are starting to lighten up. People are heading for the beach.
And it's in a crime scene truck,
Okay,
and it's there for 7 hours
okay,
before opened.
okay.
Would you agree that putting swatches in a plastic bag, wet blood swatches, in that fashion would have the effect of degrading the DNA in those samples?
Provided that there was some bacterial action, for example, that came with the substrate material, yes, that could get that process going.
And the bacterial agents could be on, you saw some pictures on direct examination about the locations of items 47, which was photo number 117, 48, photo number 118, 49, photo number 119, 50, photo number 120, and 52, I think, photo number 122. You saw those?
Yes.
Those blood drops, right?
Yes.
You saw the substrates.
Yes,
Would you have a reasonable expectation that those, that swatches made from those blood drops would contain biological, bacterial agents that when put, those swatches put in the plastic bag, kept there for 7 hours, moist and wet, would begin a process of bacterial degradation?
yes. In other words, if this is a blood sample, then the bacteria would feed on the blood, and yes.
In your opinion, is a sound practice to collect wet blood swatches of this kind containing from substrates that have some dirt, put them in plastic bags, the truck in my hypothetical, for 7 hours? Is that a sound practice?
I don't think it's a good idea to leave them that long.
Because that could have the effect of causing severe degradation of the samples.
Well, it could certainly lead to degradation. The severity is, I'm not sure of.
Well, because it's hard to predict exactly how extensive the process of degradation might be.
Well, I don't know if it's that hard to predict. I mean, bacteria have certain growth cycles, and people have characterized these things quite well. So I think you could do the experiment. I think you just don't know exactly how much is in the starting material. That's what would be hard to predict.
You conducted yield gels of the blood drops from Bundy.
Yes.
And the result of your yield gels indicated that they contained lots of bacterial DNA.
Well, that's what it suggested, although one should keep in mind we did not do a specific test for bacteria, but that's what I interpreted to be the most likely cause.
There, in these yield gels, I think that, did you see that part of Dr. Cotton's testimony where she put a yield gel up there for the jury?
I may have seen that, yes. I think I did.
The bottom of, that's one of those gels where they have some bands that are standards
Yes.
that have a certain degree of intensity,
Yes.
and then you compare the smears in the lanes for the specimens to the standards.
Yes.
And sometimes at the bottom of those bands, there's something that are called plasmids.
Yes, I think those may be plasmids, although I'm not sure those are plasmids.
Why don't you tell the jury what a plasmid is?
Yes, a plasmid is a relatively small piece of circular DNA that's usually found inside a bacterium. It's separate from the bacterial DNA, but it's goes along for the ride with the bacterium.
And on the yield gels, one can identify areas that look like plasmid bands.
Well, the point is, I know the bands you're speaking of, and I believe they are of bacterial origin. Whether they're plasmids or not, I'm not sure.
But those bands indicate to you that there was substantial bacterial degradation in the Bundy blood drops.
Well, that would suggest to me that there's substantial bacteria. For example, when we look at a vaginal swab from a case, you'll frequently see bacteria, but that doesn't mean that the female's DNA is degraded.
Now, after the yield gel for these Bundy drops, you conducted something that you described before as a southern transfer process.
Yes.
And that was done to determine if you could identify the amount of human DNA that were in these samples.
Yes.
And and you were able to get some, and then you did what's known as a slot plot.
Yes.
Did you do that before? I think you did that before or after the southern transfer.
On one of the times we did it after, I think another time we may have done it before.
But, finished? But the slot plot is a way of determining the amount of human DNA
Yes.
in the specimen.
Yes.
And so what we had in these Bundy blood drops was a substantial amount of bacterial DNA
Yes.
and some amount of human DNA.
Yes.
And from those tests, you can't tell how the amount of human DNA got there.
That's correct.
Now let's talk about how much human DNA you were able to identify in these samples.
Okay.
Now, for sample 47, you received 2 swatches.
I believe that's correct, but I'd like to check my notes on that point.
All right. If I may approach the witness, I have a chart that may assist him.
All right, you want to show that to Mr. Harmon? Mr. Scheck? Mr. Scheck?
It's my own work notes.
Well, if you're going to show it to the witness, you have to show it to...
Oh, yeah, that's fine.
Can I get a copy of it?
Sure.
I've never seen it before. Can I have a chance to review it too, Your Honor?
Sure. Mrs. Robertson, you want to...
You know what, Your Honor, just to save time rather than do that, he's going to look it up. You're going to look this up. You're not going to take my word what's on this chart, are you? You're going to look it up in your own notes?
Mr. Scheck, I'd take your word for anything.
Well, why don't you just
I'll look it up.
look it up?
But just to make sure we're connecting here. Now, this was with regards to number 47?
47.
Okay. I had, by the way, prepared a little spreadsheet that just is a roadmap to my notes. That's what I'm looking at to find these
Maybe I
things.
could look at that.
Sure.
Okay. So you received 2 swatches.
Yes.
And they weighed 8.5 milligrams.
This is now number 47. Okay, that would be correct.
And the weight extracted by you was 4.7 milligrams.
Yes, I took the larger piece.
And you got 4.34 nanograms of human DNA.
I'm sorry, you asked me how many, what was the quantity?
4.34 by the slot plot.
That sounds about right. I had about 3.8 after I did the quantitation. When Dr. Blake and I would review these quantitations, I went back and figured out he went back and figured out the total yields, I believe, but what my notes would reflect was probably how much was left after I had done the quantitation, because that's what I had left to work with. So that sounds about right, about 4 nanograms.
Yeah, 4.34.
I'd go for that.
Now, let's assume that there were 5 other swatches associated with sample 47 weighing 28 milligrams. And let's further assume that the biological matter, the blood, was randomly or evenly distributed over those other swatches.
That's quite an assumption sometimes, if you're talking about
Just
uniform.
assume that.
Okay.
Objection is improper hypothetical, there's no foundation for it.
And let's further assume that, maybe this will be the easiest assumption to just make and quantify. Let's just assume that out of all the material associated with sample 47, all 7 swatches, you extracted 12.9 or 13% of the DNA.
Okay.
Would you project then that associated with all the swatches, there would be about 33.6 nanograms?
Well, without a calculator, I wouldn't assume that, but.
About in that area.
Okay, say again, your amount was?
That you had about 12.9 or 13% of the sample,
Okay.
that if we were to calculate, thank you, Your Honor, if we were to calculate the total amount of human DNA in the sample, it would come to something on the order of 33.6
Well,
nanograms.
yeah, that sounds about right.
All
It'd be
right.
about 10 times what I had, so, yeah,
Now let's try sample 48.
okay.
You received 2 swatches weighing 8.1 milligrams. Extracted 6.5 and got a slot blot of 4 nanograms.
Again, let me check that. Okay, this is item number
48.
48. Okay. And you said about 4, is that right?
4.
Okay.
So, based on the weight, assuming that you extracted 80% of the total amount in those 2 swatches, that if you were to project total amount of DNA on the swatches, it would be 5 nanograms.
Yeah, I can eyeball that.
All right. Let's turn to sample 49.
Okay.
You received about 2 swatches and I think something like a thread that weighed 4.6 milligrams altogether.
Well, now keep in mind on 49, there were 2 different submissions on that, but you're talking about the initial one.
Yeah. Well, to cut to the bottom line, let's assume that there were altogether 5 swatches
Okay,
associated with this sample, weighing a total of 19.6 milligrams. You extracted 2.5 milligrams and got a quant of 0.24 nanograms, which would mean that your quantification represents 13% of the total sample extracted, and therefore the total amount of human DNA in the sample would be on the order of 1.8 nanograms.
that sounds about right, yes.
Let's turn to item 50.
Okay,
You received one swatch weighing 7.2 milligrams.
okay,
Assume that there's a total of 4 swatches with a total weight of 22.2 milligrams.
okay.
The weight of your extraction is 3.6.
Wait, I'm sorry, back up, there were a total of how many swatches?
4,
4. Okay, and mine was 7.2?
right.
Okay,
And the total weight is 22.2 mil.
oh wait, if there's 4 and I've got 7.2,
Mhm.
doesn't it have to be closer to about 30 milligrams?
Is this a hypothetical, or if it is, there's no foundation for it? It's
No.
a proper hypothetical.
Well, it's a little late now that we've gone through several.
Well, better late than never, Your Honor.
That's true.
Uh,
Why don't you re-ask the question, clarify with Mr. Sims?
Why don't we just, the weight you extracted was 3.6 milligrams.
Oh, okay, what I extracted was 3.6, okay, that's where,
And
Okay.
assume that's 16% of the total.
okay.
Is that a hypothetical? If it
I'm
is,
asking him
there's
to
no-
assume that.
There's no basis for it. Objection on that basis.
Sustained.
Your Honor, you're aware that all the swatches were weighed in this case at your laboratory at Cellmark Diagnostics.
Objection. Calls for hearsay, it's compound.
Sustained.
Your Honor, as subject to connection,